So, see here. Wait, I will send in the that Zoom meeting. Wait. So where is the Zoom? So wait sometime here. Okay. No. Okay. So these techniques are here. So these techniques are there in the semiclonal somatic means micropropagation. Okay. In the stage zero, what happened here? The initial step is micropropagation is the stage that one we call as the zero stage, which is involve the selection and growth of the stock plants. That is means mother plant. It is considered around the 3 months under controlled condition only. The stock plant means mother plants we have to choose, and which is the around 3 months plants are there, which is under control. Okay, so and the stage one. In the stage one is initiated, establish the culture in the suitable media, selection of appropriate explant is important. The most commonly used explants are argon, shoot tips, auxiliary boards. The chosen explant is surface sterilized and washed before when we use. And stage two, the major activity of micropropagation occur in differentiated culture medium. The stage two is mainly involve the multiplication of shoots and rapid embryo or callus formation from the explant. And stage three here, this one is involved, the shoot is medium and rapid development into roots and sometimes shoots also developed. The in vitro rooting of shoot is preferred while stimulating the handling a large number of species. Another, that is the last stage, fourth stage is there. In the stage involve establishment of plantlets in a soil. This is the done by transferring the plantation of stage. The micropropagation mostly involve the in vitro clonal propagation by following the approaches are there. This auxiliary bird proliferation, organogenesis, somatic embryogenesis. What is the auxiliary bird proliferation? See here, auxiliary bird proliferation. So auxiliary bird proliferation is okay here. So auxiliary bird proliferation. The meristem are present at the axillary, that is the shoot tips, either root tips only sometimes. The stimulation of axillary birds, which are usually present in the axil of each leaf to develop into the shoot. In mature, these birds remain dominant for the various period depending upon the growth pattern of the plant due to continuous application of siloxane in the culture media shoot formed by birds which is present on the X plant. So So, okay. So somatic embryogenesis. Other one is the somatic embryogenesis. I on to this picture here. Hmm. Somatic embryogenesis. See here. Hmm. Okay. So what is the somatic embryogenesis? Means the regeneration of embryos from somatic cells. That is the somatic cells are gone. This one is the somatic embryogenesis may uh, result in non-zygotic embryos or somatic embryos. There are the directly from formed from the somatic organs. This is the another one is there in the here the somatic embryogenesis. Here two types are there: direct to embryogenesis. Another one is the indirect to embryogenesis. The embryo intake directly from the explant without callus formation called the direct embryogenesis. Means there is no callus in the direct embryogenesis. And here some cells which are called as pre-embryonic determined cells are initiated embryonic development. Such cells are formed mostly in embryonic tissues contain that is a certain tissues of young in growth plant and hypocotyls, etc. So indirect embryogenesis, the embryos are developed through cell proliferation that are called callus formation called indirect embryogenesis. So that cell proliferation they are formed the callus in the indirect embryogenesis. The cells from which embryos arise are called as inductive embryogenesis determine the cells. For this growth regulates with a specific culture conditions are so for the growth regulates and the specific culture condition are required for the callus and then is the determination of those cells into the embryo development. Okay, this is the embryogenesis and some. Okay, now we are discuss about the block 2, that is in vitro culture. In vitro culture, here in vitro morphogenesis. What we are discuss about the in vitro morphogenesis. The meaning of the term morphogenesis, which are all activities involved in the formation of a complete individual, starting from the cells or tissues in culture. The various internal as well as external factors affect the morphogenesis potential of cells. The morphogenesis is always also may be termed as a development morphology. What is the definition of morphogenesis? Means the branch of biology that deals with the uh, causes and activity during the organization of complete individual plant. This is the morphogenesis. So, anatomical and physiological events which are involved in the growth and development of an uh, so here there are the some factors are there in the morphogenesis. Okay. The morphogenesis, here some are the factors. What are the factors are there here? Discuss. There are many factors which influence the morphogenesis potential of cells by affecting important developmental activity. These include the experiments have shown the certain growth factors like cytokinin include the somatic embryos in culture by changing the cell polarity. Some chemical factors which are promote the asymmetric division, the cultural cells, those testing the difference in the form. The variation concentration of 2,4-D, 2,4-dichlorophenic acid, 2,4-D, that one, 2,4-D, auxins, all these development of organ in culture by the causing the alternate cell elongation and the ratio of cell division. With the increase in culture is the duration of the organogenetic differentiation show this decline. All the the different factors have the different type effect on different developmental activity. So many factors having a great importance is deciding the morphogenesis pattern. There are the physical factor like light and temperature, pH. So chemical factors like carbon, nitrogen ratio, oxygen content, and carbohydrates, the concentration of carbohydrates. These are the some chemical factors. Growth hormones like auxin, cytokinin, gibberellins are there, and water culture duration also. Mechanical strain and pressure depending on bending in many cases, the plant cell division that may cause the differentiation morphogen potential. So gene expression changes in the cells in the culture and electrical stimulation, genotype, and its status of nucleic acid in different cells. Repeated substructure of the totipotency callus may also result in the reduction or completely in the loss of morphogenetic potential. Terminology, what are the terminology here? Totipotency. The cells have the ability to regenerate into plantlets on the particular medium. That character we call as a totipotency. So cellular is a differentiation, the morphogenesis development of higher plants. Another one is the oxalogenesis, differentiation of parenchyma into localized secondary wall thickening in xylem tissue. Okay, and see here. So organogenesis, in vitro generation of shoot, root, floral buds from explant. That is called organogenesis. Already direct and indirect organogenesis that already discussed. Somatic embryogenesis. The somatic embryogenesis is the synthetic seeds in vitro regeneration of somatic embryos from explant. Somatic embryos is an additional regeneration pathway exclusive to plant system. Encapsulation of embryos with the hydrogels. So this is another here. What is that one here? Encapsulation embryos with the hydrogels. That is redundant. The pioneered use of hydrogels for the encapsulation seems to be one of the promising method for showing the embryos in the field. So the encapsulation with the proper material and structure will not only protect the embryo from the physical damage, or sometimes is the discussion during the delivery or some owing the process in the greenhouse, but also enable easy handling and automation. And so here another one is the androgenesis. Androgenesis, what is the androgenesis? In vitro differentiation that callus are plantlets from microspores. Which is isolated from the pollen grains by culture of exide anther. That one is the androgenesis. In the homozygous diploid can be raised from these applied by the colchicine treatment. The colchicine, you know that one is the colchicine is a spindle fiber break the is chemical. And another one is the gynogenesis. In vitro generation, that is the regeneration of applied plants from the unpollinated female gametophyte, that is embryo sac, by culturing ovule known as the gynogenesis. This one is the first discovered by the here San Num. So another here we are to discuss about the types of cultures, and tomorrow we discuss these types of cultures are there. Here types of culture, that is a cell culture, organ culture, callus culture, and protoplast culture. Here are different types of the cultures are there. Okay, so just wait 10 minutes. There we are discussing about these types of cultures. Here there are the different types of plant tissue culture technique, mainly based on the explant used. So based on the text plant, which part we are to use it as the source of the culture, that one we are categorized into different cultures are there. The cell culture, organ culture, callus culture, protoplast culture. This cell culture is the culture is isolated in individual cells obtained from the explant tissues, or either callus is regards as a cell culture. Sometimes ovules also there, anther culture, endosperm, these are also there. What is the cell culture? Okay, see here the types of I shown to these uh, types of cell culture means here. So types of culture: cell culture, organ culture, callus culture, and protoplast culture. The cell culture culture of isolated individual cells obtained from an explant callus or tissues is regarded as cell culture. So these culture are carried out the dispersion medium and referred to as the cell suspension culture. This culture is a single cell was made in early in Haberlands. Isolation of single cell. Isolation of single cell. The the cells employed for in vitro culture may be isolated from culture tissues or plant organs. The single cell can be isolated from callus culture. Repeat subculturing of callus on agar medium improves the variability of callus, so that the free cell suspension are obtained. The plant lives with the homogeneous population of cells are ideal source for the cell culture. Cells are isolated through the mechanical method or enzymatic method. The cell culture technique here suspension culture. The isolated cells are grown in suspension culture. The cell suspensions are maintained by return subculturing. When a new suspension culture is started, it necessary to determine the okay. Another is there. What is the here? There are difference into two types are there in the suspension culture. What is that one is the batch culture, another one is the continuous culture. The batch culture is grown in the fixed volume of nutrient culture media. In this culture, the cell division growth complete with the increase in biomass occur until one hour factor that is the culture environmental became limiting. The cells exhibits the forming the five phases growth when the cell number in the suspension culture that is the long phase, log phase, and linear phase, declaration phase, stationary phase. Log phase characterized by the preparation of cells to divide. That is the log phase where the rate of cells multiplication is highest. The linear phase it is the represented by uh, slowness in cell division and increase in cell size expression. That is the stationary phase is represented by the constant number of cells and their size. Fourth one is the declaration phase. It is characterized by the decrease in cell division and cell expansion. Fifth one is the stationary phase. It is represented by the constant number of cells and their size. The batch culture can be maintained by the continuous transfer of small cells. So another one is the continuous culture. In this culture, there is frequently additional fresh medium during the out of the medium. So that is the culture volume is normal constant. This culture is carried out the specially designed culture vessels. Continuous cultures are two types here: cloned continuous culture and open continuous culture. What is the cloned continuous culture here? In this culture, these cells retain while the influence of fresh medium is balanced with the outflow of the corresponding spent medium. Open continuous culture. In this culture, the inflow of fresh medium is balanced with the outflow of the volume of spent medium along with the culture. The additional of fresh medium and culture harvest are adjacent that are the culture are maintained the identified at the constant growth of. So another here synchronization of suspension culture, physical method and another chemical methods are there. And here physical method, chemical method, here discuss that one. And finally the staining technique. After we are to discuss staining technique, see here this is the staining diagram summary of the cell suspension culture. I shown to this picture. See here. So this one is the shown suspension culture. See here. So this is the diagram for suspension culture. Okay. This is the suspension culture. Okay. Um, Suspension culture. So here the additional liquid medium here. After that liquid medium, then micro chamber cell culture. Then callus will form. Then callus again they transfer to sub-boils. That's regeneration. And after plantlets, there are transferred into the that is the small pots and then shifting to the directly into the field. This is uh, the suspension culture. Okay, so we are thanking you for attending the class and thank you so much to attend the class. Okay, thank you everybody. Tomorrow we discuss with the another topic.